Hello, everyone. Thanks for joining us today. I am going to go through our order of ceremonies a little bit today, and I assume that John's going to put my presentation over here for me in a minute. I'm going to let him do that, though, so I don't mess it up. But the Science of Soil Health Initiative was something started from the interest that's been developed and training that's been developed around soil health within NRCS and really across agriculture. And so as this topic has really come to the forefront, people wanted to know, what does this really mean? What are the real science and values that are behind things? So I put this information into something I call DSPs for soil health. So we sort of organize things around a common set of projects. So the way this is going to work today is we're going to have a lot of cooperators present a little bit, just a little bit about their projects, and I'm going to be fairly severe about keeping people on time just so it doesn't take too long. So then once all of those overviews are over, we'll get to your questions and answers, and we'll have each individual presenter speak to those individually. So if you can put your questions into the Q&A pod as we're going through it, or maybe you've watched the pre-recorded presentations beforehand, if you can address your questions specifically, that'll help us sort through who you meant those questions for and make sure you get the right answer. There's a lot more information on the website, on the flyer that John put up initially. The links are there. We'll make sure you get that information, and there's a lot more information to be had. So I'm going to start with a general overview of the entire project. So Science of Soil Health Initiative, as I said, we want to know what the real science is behind soil health in a lot of different areas. And a lot of people, when they do soil health projects or they talk about what soil health means, they really target it for their specific areas, which makes sense, right? Because that's how you manage the landscape. So if you want to know what a good soil health value is, you really kind of have to know where it's from. And so we put together this big project we call Dynamic Soil Properties for Soil Health. So we're measuring things that soil survey thinks of as dynamic soil properties, but we really have an eye towards getting a handle on soil health. And we have 18 agreements across the country that use a pretty similar setup. So what we want to do is assess a common set of laboratory and field indicators for a range of soils and a range of management systems or conditions. And we have several goals that we're trying to do at the same time. Some of them more successfully than others, but we want to develop reference values so we know what good and bad or okay is for any given indicator in any given soil. We want to assess usefulness of soil health indicators in individual circumstances, so maybe some of these properties don't really work that well in certain situations. And then we want to link research to soil survey databases. So for each of these projects, there was a soil survey characterization kit component to it. We're not going to be talking about those so much today, but in the future we will be. And then finally, we want to compare results across these different labs and with some stuff done at KSSL just to see how repeatable it is. Oh, somebody needs to mute, I think. Okay, so there's a bunch of cooperating institutions, as I said, like 18 of them. Oh, and I should have mentioned we have a hashtag. That's why this DSP for Soil Health hashtag comes in on Twitter. And a lot of people have already posted things if you're interested in looking at that or sharing. So we'll start out today with NC State with Caitlin Cottle. Would you give us your quick 3-minute overview of these 3 slides and just let me know when you want to go to the next slide?
Sure. So my name is Caitlin Cottle. I was a grad student that worked on this project, and we focused on soil health between 2 different management systems, and that was the crop and hay systems. You can go ahead, Ty.
Okay.
So all of our fields were MAP Cecil, which is the common soil all across the southeast U.S. We had 3 crop fields, 3 hay fields, and 1 forest field to serve as an undisturbed soil. We dug 1 pit and 1 field of each system for the characterization. We had 3 sample locations in each field, all oriented down slope, and these sample locations were equidistant apart within the field, and they were about 15 feet from the edge of the field. We had a total of 20 sample locations, So we had 9 crop sample locations, 9 hay sample locations, and 2 forest sample locations. Again, go ahead. We only had one metric that was able to detect a difference between the systems, and that was soil organic carbon. Most metrics were able to detect a difference with depth, but the significant differences between the systems was typically at the 0 to 5 centimeter depth. Metrics meant to represent similar soil properties showed different interactions or trends, and that was typically with the biological properties. Many of the metrics were significantly correlated. Some of the protocols were a little hard for me to understand in the beginning, but we worked through that with NRCS, and a lot of the protocols are pretty labor intensive and time consuming, so they might not be suitable for high throughput settings.
All right, great. You hit it right on time, Caitlin. Okay, so our next presentation is from Kansas State, and that's Peter Tomlinson, if he's on, or Deanne, if she is still around. Peter, are you here? Yes, go ahead.
Okay, all right. I am running a bit blind as I'm still not successfully into Adobe Connect, but so at Kansas State, we focused on the Keith Soil Series as our benchmark soil, mapped predominantly throughout western Kansas. And so we did our sampling in the fall, and so Sky, if you can go to our second slide.
Okay.
I'm getting them open here as well. We'll get there.
Technical difficulties.
Technical difficulties, yes.
I can help out, Peter. I'm still here. I'm not teaching class yet.
Are you there?
So, yeah, I'm here.
Okay, go ahead, Deanne.
So, this is a schematic from one of our sites. So in the lower corner or at the bottom of the map, you can see, well, first of all, in the inset map, you can see what part of the state we were working in. And then this is one of our particular sites in the photo. So we had our no system with diverse crop rotation in the points at the top of the map. So in each field, and we try to stay on similar landscape positions. We pulled 3 cores, so those were sort of replicates. And then down in the bottom of the picture, you can see where we were in a prairie remnant, which this soil is so productive that prairie remnants are not common at all. So, and we also went with dryland because this soil is very productive when irrigated, but you're not going to have any irrigated native prairies. So we wanted to keep it the same, so we went with dryland. And so next slide will be our results. And so I just pulled together some of the different properties here. So as far as water stable aggregates and respiration, beta-glucosidase, and the phospholipid fatty acid total weight, the trends all fell the same, which is that conventional conventionally farmed soils, which we defined as conventional tillage and pretty boring crop rotation, those grouped together with our soil health systems. So the ones that were managed with the diverse crop rotation, no-till for longer than 20 years, cover crops for 9 years or so, and the rangeland was significantly different than those farm systems. So that's about all I'll say on that. So in other words, we didn't have very many properties where the soil health managed system had distinguished itself from the conventionally tilled system yet. Okay, great. Thanks. Sorry for the technical difficulties, guys. Thanks for stepping up. So for the next presentation, we'll have Joseph Burke from Texas A&M AgriLife.
Hi, everyone. Joseph Burke here, Texas A&M, but we're based in Lubbock for this presentation. Co-presenters are Drs. Katie Lewis and Paul DeLon at both the Lubbock Research Center and Dr. DeLon in Vernon. You can go to the next slide. So one of the key things about working in West Texas is the fact that the Dust Bowl is remnant throughout all of the work that we do. So the picture on the top left-hand side is from Hansford County, far north Texas Panhandle. The bottom picture, and that was from the Dust Bowl itself in 1935. The picture below it is from Terry County in 2013, and that is just adjacent to one of our major research sites. And so what you can see is that we're still having issues with dealing with wind erosion and erodibility on the Texas High Plains. And that dominates how we look at conservation systems. Cotton is also king here. And so we have two research sites that we looked at that have predominant series of the Amarillo fine sandy loam in the south and the Pullman clay loam to the north. And those are represented by those yellow boxes. You go next slide. Most of our results were similar for all of these. They didn't transfer very well. I apologize for that. So we have four figures that we're showing: soil organic carbon, total PLFAs, beta-glucosidase, and beta-glucosaminidase. And what we see is that with each of our treatments, and if you look at the presentation on YouTube, they're more clear than they are here. With our conservation practices, both the rye cover crop and the mixed species cover with no tillage, and the native site that we compared back to are far greater in their soil health indicators compared to the conventional system. And one of the questions that our farmers continuously have is, how does this relate to yield? What we see is that there's no relationship between the biological indicators for soil health and cotton yield on the semi-arid Texas High Plains. Still looking forward to any questions if you might have them, though.
Great, thanks. All right, we're going to swing to the whole other coast of the country for Washington State. I'm pretty sure Hyene was on, but I'm not sure if she was able to call in. Well, I'm not going to explain this very complicated slide poorly, so we can come back if you're able to get onto the phone here in a moment. So James, Lynn, are you on? We'll go to the other K-State.
Yeah, I'm here.
Oh, hi.
So for
Go ahead, James.
Okay. So I'm a master's student with Kansas State University under Dr. Charles Rice. And for my project on the soil health metrics research, the objectives for the project was to study the soil health status across a Kansas precipitation gradient under 3 different long-term land management systems. So the 3 different systems I'll go over in a little bit, but using those, I also wanted to assess the utility and reliability of the standard set of soil health procedures given with different soil types down to a 100-centimeter depth. And then there's also different methods that are generally used, like standard methods by my lab, so I also wanted to compare those methods with the standard set given to me. Next slide. So for the different long-term land management sites, I had native prairie, which varies by tall grass and short grass from east to west. I had conventional tilled agriculture, and I had enhanced agriculture, which was a variation with no-till and either crop rotation or cover crops. For the soils I had, or the locations of the soils I had, were located at Tribune, Hayes, Conza, which is also Manhattan, and Ottawa. And as you can see, the precipitation increases from west to east. Each soil type were fairly similar or the same for each land management system. Next slide. So this is just an overall soil health sample intake and processing overview to show the different methods that I had to go do with each set of soils. You can see the results on the YouTube presentation I have, but in general, we can see for the results, basically that native prairie had greater soil health than the ag sites, and then only at Hayes did we see enhanced agriculture having better soil health than conventional till. I'm still in the process of analyzing the data, so that'll take time, but thank you.
Thanks, James. Next up, we have Regina O'Kelly from Oregon State University.
Hi. So, hello, I'm Regina. I'm a PhD candidate working on the Willamette Valley, Oregon DSPs project. My advisor, Dr. David Myrold, is the principal investigator for this project. During this virtual symposium, I wanted to take a more focused approach and ask a couple specific questions about how the carbon properties fit together. Next slide, please. So we sampled from a diverse range of perennial management systems on 2 soils in the valley, a mollisol and an aldosol. In the suite of dynamic soil properties, there are a few different ways to assess the available carbon resources for microbes. Soil organic carbon with dry combustion is one. You can also measure mineralizable carbon, also called microbial respiration, and permanganate oxidizable carbon, or POXI, which will be my focus today. POXI is a popular, sorry. Thanks.
Sorry, operator error. Go ahead.
So POXI is a popular soil health indicator. Studies have found that it is sensitive to management, and it has been reported to be representative of a readily accessible fraction of carbon for microbes. However, studies have not universally supported these claims, so I wanted to look at how well they held up in the Willamette Valley soils. So for this second claim, that POXI is a particularly active part of organic carbon that microbes are using more, if that's true, then I would expect that it should be more closely related with respiration or mineralizable carbon. Next slide, please. So for our results, with respect to the claim about sensitivity to management, we did not see very much difference between management systems for POXI. However, POXI was fairly well correlated to respiration and soil organic carbon and followed the same trend between management systems, even though there weren't significant differences. So looking at these 2 figures, we have 2 different ways to explain mineralized carbon, which we can think about as an index of microbial activity. So POXI is on the left and soil organic carbon on the right. And based on these R-squared values, soil organic carbon is more tightly correlated with mineralized carbon than POXI was. And so this does not suggest that POXI is a particularly active fraction of carbon. So despite POXI not quite matching with some of these claims, I still see POXI as a relatively inexpensive and quick way to estimate carbon resources, which makes it a valuable dynamic soil property for soil health research. Thank you.
Great, thanks, Gina. Next up, we have Susan Crow from, I'm going to let you say it because I will not pronounce it correctly. Go ahead.
Thanks. Susan Crow from University of Hawaii, and acknowledgments to my co-PI, Jonathan Dinick, and Elaine Giska, who is the PhD student who will work on this project. We have not started yet, so this is quite different from the other groups. But next slide, please. Hawaii's diverse soils and ecosystems make it really challenging for us to accurately represent them in decision support tools. So our group has surveyed a range of productive systems in Hawaii, and we developed from that a proposed list of soil health indicators specifically for subtropical tropical systems, and also identified that mineralogy and land use history are key factors in determining soil health. So andisols, or volcanic ash-derived soils, have the second greatest extent and are among the most fertile soils in Hawaii, and we've observed that intensive cultivation reduces some common indicators of soil health, including total organic carbon, CO2 burst, and water-stable aggregates, but these systems are generally understudied in pursuit of soil health knowledge and basic understanding. Next slide, please. So the objective of our project, which is just starting, is to identify differences in soil health status across an environmental, specifically climate gradient, under different land uses and using standard sets of field and lab methods in Hawaii. And so we will focus on a climate gradient that resulted in 2 different andisols forming with contrasting properties, eudic and ustic regimes. So for each andesol, we'll measure soil health status in undisturbed, intermediate, and intensively cultivated sites, which we are picking next month. And we will apply standard protocols from NRCS as well as our regionally developed protocols to ensure that data harmonization and translation across metrics. So our goal generally is to help develop region-specific knowledge of the science of soil health that is required to recommend best practices moving forward to promote resilient landscapes, sustainable management, and climate change mitigation. Thank you.
Right, great, thank you. Next up, we have a joint project from Alabama A&M and University of Maryland Eastern Shore. Diedrich, were you able to get on the telephone? It sounds like maybe not. So that may be another person that we have to loop back with and join up at the end. We'll move on to University of Illinois. Mario or Carmen, I'm not sure who's presenting. Carmen?
Hi. Yeah. Good afternoon. This is Carmen Ugarte, and what I'm going to be presenting is some work that was started by my postdoc, Peter O'Bour. And Mario here, who's my co-author, a visiting scientist from Honduras, who took over the heavy lifting after Peter got a faculty position. So our project is looking at grain cropping systems in Illinois, and we're working with collaborators that farm using non-till, organic, and conventional practices. And we also had the opportunity to sample from an area that has been restored and is near the University of Illinois. So in total, we had 7 fields. And in my next slide, what I have is more focusing on, Sky, if you could go to the next slide, I'm looking at 2 of those cropping systems. We're looking at the conventional and organic systems, and in terms of management, they are characterized by the rotation length, by the sources of fertility and organic matter inputs, as well as the frequency and intensity of tillage practices. On the right side of the slide, you see a schematic of how we originally planned our sampling. We distinguished what we call management zones that are based on landscape characteristics, drainage classes, and satellite imagery. Once in the field, we had to readjust our original sampling plans to be able to control for the specific soil types that we were interested on. So we are looking at the Drummer-Flanagan Association here. In the next slide, we have data on soil organic carbon, permanganate oxidizable carbon, and beta-glucosidase. And we found, just not very surprisingly in that sense, that epoxy and beta-glucosidase were more variable than SOC, suggesting that if we're going to use these as a more readily available indicator, we may need to account for a more intensive sampling protocol so that we're able to account for this variability. Looking at the trends, the results are also in agreement with a previous meta-analysis that we conducted, and that suggests that organic cropping systems tend to accumulate more organic matter in the subsoil. Likely this could be influenced by the use of cover crops and relatively more diverse rotation sequence. And we're eager about contrasting these results with the non-till system data that's currently being under processing. And also we're looking at comparisons of other indicators that we have been working with. So thank you.
Great, thanks, Carmen. Okay, next we have, I'm not going to butcher your name, Luz is what she goes by, from the University of Texas, Rio Grande Valley. and she's going to tell us about a project they're just getting started on down there. Go ahead.
Okay, yes, no worries. Thank you. So my name is Luciana Gonzalez. I go by Luz. I'm a master's student at the University of Texas, Rio Grande Valley. And you can go to the next slide. Perfect. So we are currently in the process of sampling. So we have a total of 11 sites. We decided to go ahead and sample from the fields of a partner farm with the agroecology lab. So we have 3 conventional tilled fields that we are sampling, 3 strip tilled fields, and then 3 intermittent tilled fields. The intermittent were previously under conservation tillage for 3 years, but then as of last winter, they became conventional. And so the way we're doing it, we didn't use a geoprobe. We're using an Edelman probe. And so we're taking 2 1-meter deep cores, and then for the first top 10 centimeters, we're using those cores that you see on the picture. In addition, we're doing the infiltration, like most of us with the single ring, the Cornell, but we're also adding the Saturo infiltration readings. And then we have decided to kind of split this project between myself and 2 other grad students, which you can see the picture of us working on the field. So essentially we decided to split this project into the chemistry, the physics, and the biology. And then another thing that I want to note is we're looking at specifically only Hidalgo sandy clay loam. And then we have the 2 ecological references. And then you can go to the next slide. And so we haven't, we are in the process of sending all of our samples and testing all of our samples. So these are what we expect to see, which is I think what some of you have already presented on. So higher steady infiltration rates and sites under that conservation tillage, soil biological activity, also improved under the strip tilled conditions, and then higher soil organic matter for those under conservation tillage. And then the end.
Great, thanks, Luz. Oh, go ahead.
Thank you.
Thank you very much. All right, now we're going to go a lot farther north up to Minnesota for Sharon.
Hi everyone, I'm Sharon Perron. I'm a PhD candidate at the University of Minnesota, and I've been coordinating the laboratory component of our DSP contract. So I'm going to start with an overview of our project and take a few minutes to just reflect on our general observations thus far. Our project takes place in southeastern Minnesota in MLRA 104, and we're investigating the tax-adjunct series Reedland, Marquis, and Kenyon, which are all mollizols that formed in a transitional ecosystem bordering tallgrass prairie and oak savanna, and typically have pretty deep A horizons, as you can see. Our sampling strategy was to sample from the same landscape position in areas marked as Reedland on the web soil survey, but ultimately we ended up describing these 3 series from our samples, which range in their drainage class. So you'll see that Reedland is the most poorly drained soil, and Kenyon is the most well-drained soil out of the pedons that we collected. Together, these series represent a little less than 1 million acres in southeastern Minnesota and northeastern Iowa. And if you look at that map there, you see a little blip of red in southeastern Minnesota there, south of the Twin Cities, and that's where we're operating. Next slide. We sampled from 3 management systems, which occurred on an approximately 400-acre section where our farmer collaborator owned the entire property, and he farmed half while renting the other half. So the property that was rented was a conventional property with what's a pretty standard corn soil rotation for this region. This farmer performed spring and fall tillage, sometimes up to 12 inches, so fairly deep, and also used herbicide and fungicide. In the alternative treatment, we had a more variable rotation with corn, soybeans, cereal rye, oats, and crimson clover just in the last few years. With minimal tillage, there was one tillage event in the last 6 years and no additional fertilizer in the past 6 years. Our reference site, we had to make do with a fence row that has never been tilled in recent memory. The vegetation there was mainly giant ragweed, stinging nettle, and brome grass. And even though it was considered sort of this reference site, there is evidence of disturbance there. So there's about a 4 to 6 inch silty surface sediment deposition in that treatment. And we sampled in fall of 2019 at the end of a soybean season in both agricultural fields. Next slide. Okay, a couple of quick takeaways from our project. We're finding it a little bit difficult in our analysis to understand what some of the true differences between the agricultural treatments and the reference treatment are due to some fairly severe erosion in some of those agricultural sites. So we found evidence of both wind and water erosion at the site, and we're trying to get sort of the most accurate estimate of deposition and see if we can adjust our analyses accordingly, but doing that accurately is going to be fairly difficult. Number 2, the methods outlined in the NRCS technical note generally work really well for our soils. We have really wonderfully loamy and rich soils in this region of Minnesota, and so far we've gotten pretty precise replicates and clean data from these methods, which is good. That said, thus far we haven't really found much evidence that these methods are distinguishing amongst our treatments, and I hypothesize it's likely due to the very high organic matter content in these molecules, as well as the fact that our alternative agricultural field actually has been farmed conventionally for several years, and really this sort of transitional ecological management has only been performed for about 6 to 9 years transitionally. Third, we're seeing very significant differences between our reference soils and our agricultural soils, so to some extent we can see that we're not reaching full capacity of these metrics in our agricultural soils. Number 4, the alternative treatment had a higher bulk density than our conventional treatment, which is fairly consistent with previous research from our region and the observation documenting that compaction during a transition period to no-till. So I would like to play around with that data and see if we can distinguish differences between treatments when accounting for bulk density, but I haven't yet been able to do that and understand how that analysis might be eliminating. And finally, number 5, soil handling and processing can have a pretty profound effect on certain methods. We all know this, that's nothing new. For a particular example, with aggregate stability, we found that our ARS method did not distinguish between agricultural treatments, but our KSSL method was able to distinguish between the conventional and alternative treatments, where the alternative treatment had a higher stability. So that was quite a lot. I'm happy to answer any questions in the Q&A. Thank you.
Thanks, Sharon. That's a lot of info. Now we're going to make our circle around back to the east coast of the United States and go to the University of Connecticut. Weijie, are you on the phone line?
Yes. Can you hear me?
Okay. Yes. Go ahead.
Okay. Hi, everyone. I'm Weijie Gang. I'm a postdoc working with Dr. Carl Guyard at the University of Connecticut. Today, I'm going to focus on the field infiltration measurement that we did last summer. You can, next slide. So in Connecticut here that we collect soil samples, focus on the 6 dominant management systems here, including conventional till corn field, no-till corn. We also sample tillgrass that manage as residential lawn, hay, forests that have been under active thinning practice, and unmanaged forests. We're still in the process of analyzing all the soil data, but today I'm going to show you some of the findings that we have based on the field infiltration measurements that compare the Cornell sprinkle rain infiltrometer and the single rain infiltration measurement. Next slide, please. The full graph here in the first top left graph is showing the infiltration measurement from the Cornell sprinkle rain infiltrometer, and this method uses a rain stimulator, and it measures the steady state infiltration when the soil is totally saturated with water. And you can see that as management intensity increase, there's a decline in the infiltrations, the lowest being the conventional till cornfields. And we see the same trend in the single ring infiltration data, which is the graph on the right side, the top right. But one thing you can see is that the y-axis in the single ring infiltrator is one order of magnitude higher than the Cornell single ring infiltration measurements. And we see very weak correlations between the two measurements. That's simply because the single ring infiltration measurement that just measure 1 inch of water when you put it on the ground, like how fast it infiltrates in the soil. So it does not really measure the steady state infiltration. So it's a measurement of both the subactivity of the soil as well as the infiltration process. And if you repeatedly adding more water into the single ring, and eventually it will become more saturated, and the measurement between the two measurements will converge. So for two things that we learned from this measurement, the first thing is that at glacier till soil, which is the soil that we have here, we normally have very well-drained soil. Even the lowest cornfield, we still have over 10 centimeters per hour water can infiltrate through the soil. That is higher than any like heavy rain that we receive here. So that's make us wondering whether like field infiltrations is a good like indicator to include when assessing soil health under like this kind of like soil conditions. And the second thing we would like to explore next is whether the lower infiltrations in the cornfield is actually become like limited in affecting plant growth, because farmers always express the opposite, that the soil has too high infiltrations. So in this case, maybe other kind of physical indicators such as like water retention may be a better indicator to include in soil health assessment in this region. And another thing we want to explore further is to understand the discrepancy between the two methods and whether we can find a way to make sense of the different patterns from these two methods. Okay, thank you.
Great, thank you. I'm so glad you focused on something the rest of us have kind of skipped over. So next we will move to the dry side of soil health, and I think Kirsten is going to get us started on that one.
Yes. Hi, everybody. Can you hear me well?
Yes.
Okay. So greetings from sunny Arizona, and I really mean sunny, where we get about 281 sunny days, which you all might be very jealous of at this point in time for our winter. But, when we have temperatures in excess of 107 degrees for, 2 odd months at a time, it can get a lot less fun. So as Sky mentioned, we are the dry side of soil health. We're one of the exclusively arid system DSPs projects. So as a part of our team, we have Dr. Joseph Blankenship. He is our esteemed PI, myself, Kirsten Ball. We have our doctor in training, I guess, or hopefully one day, Chance Moscarella, and our lab manager, Sam Rapke, who is not here with us today, but is an absolutely integral part of our program. I also just wanted to mention that we have a whole bunch of field sites for our program. It's going to be impossible for me to go through everything today, and we are just starting with our project, so we don't have a heck of a lot of data to show you. So I hope to kind of give you an arid soil health overview, but I just wanted to acknowledge that our sites occur on Tohono O'odham, Gila River, and Yavapai Prescott Native Tribal lands. Next slide, please, Sky. So in dry lands, we have some pretty interesting soil health challenges that definitely differ from more temperate regions. So we're finding in looking at the mixture of the physical, chemical, and biological components that make up soil health, there are a bunch of things that we're finding are going to be challenging, both for understanding how soil health works, but also in conjunction with that, how it affects the way that we actually measure our soil health properties. So not the least of which is that all of our agricultural systems are irrigation dependent. They tend to be high eroding. We either have problems with too fast or too slow infiltration, and we tend to have very low to almost nil aggregation. And I want to also say that some of the mechanisms controlling our aggregation are somewhat unclear, related to some of our chemical and biological indicators, like the fact that soil organic matter is very, very low in these systems. Inorganic carbon often outweighs the total amount of organic carbon that is in the soil, and we're dealing with very high pHs. For example, one of our sites out in Goodyear has an average soil pH of about 8.5. So we're looking at some very unique challenges chemically. In addition, we always expect that we have very low net primary productivity in our systems, even those that are under irrigation. For example, we're working in some viticultural regions, and some of our viticulturalists, no matter how much fertilizer and water they tend to dump on their fields or their cropping systems, they find it very hard to get a lot of vegetative growth. So we always expect that we're going to have a minimal amount of organic matter build up in these systems, and as a component of that, we expect that there will be very low biodiversity and microbial biomass. Those two things obviously work very intrinsically together. If you can't have a substrate for microbial biomass, then you're not going to have a growing microbial biomass, and the chicken egg scenario continues. Next slide, please, Sky. So when we started this project, there were a lot of discussions that we had around the fact that we really need a new framework for soil health assessment that is quite specific to arid systems. There remains a lot that we don't know about the systems, about what even soil health really means in these systems. For example, how much organic matter can we actually build in these systems? You know, is it even possible to get, you know, significant increases whereby the soil becomes quote unquote healthy? You can't see my air quotes. And therefore, some of the measurements that we're taking might not actually be informative. For example, we are having some concerns about using some of the biological metrics specifically, like substrate-induced respiration, which tends to underestimate microbial biomass in arid alkaline systems. And as if you saw our presentation with Chance, how maybe the soil exoenzyme measurements might be affected by pH. And so from the data that we're seeing initially, at least from these systems, we're finding some pretty counterintuitive patterns which have significant implications for management, not the least of which is that we're finding that potentially conventional tillage as opposed to a minimal tillage system in arid alkaline soils might actually be increasing soil organic matter as opposed to a minimal tillage, which would be expected in a more temperate system. So we're looking forward to sharing our future data with all of you and having some great chats. Thank you for your time today.
Great, thanks. Definitely an interesting area to work in. Okay, we're trying to move back to, let me see. Oh, I hit the wrong button. Oh yeah, there we go. So, Haiying, are you on the phone now?
Yes, I am. Can you hear me now?
Yes.
Hello? Okay, great. Thank you. So my name is Haiying Cao. I actually the talk was submitted by Catherine Nesco, but she's not able to be here today, so I'm filling her role for her presentation. So in Washington State, we took samples. I wish I put some maps on the first slide, but in Washington State, we took samples from fields that represent major crop production in dryland, wheat-based area of eastern Washington. The soil series that we focused on was silt loam series, which is a major soil type in the Pacific Northwest and one of the most fertile deep soil in the world. We took samples from 8 fields that were from 2 major agroecological systems in the Pacific Northwest. So in dryland area, we classify our environment into 3 agroecological zones. We took samples from 2 of them. One was from green fallow agroecological zone, which represents the dry area that has annual rainfall of less than 12 inches. And then we took samples from fields that are located in annual crop agroecological zone, which represents the wetter area of dryland cropping systems with annual rainfall around 20 to 29-ish inches. And some fields were on commercial farms, and some fields are long-term research sites on our research farms of Washington State University as well as University of Idaho. So the management systems that we focused on were conventional till systems, no-till systems, as well as long-term no-till prairie grassland reference, mainly perennial wheatgrass. So this represents what the soil would look like if we did not have agricultural production. At least we wish, because that strip was a production site before, but and then we put it back to prairie grass as a reference years ago. So since our fields in the Plus area are hilly, some of the slopes could be as steep as almost 40 degrees. Is really, really steep hills we have in here. We took samples along slopes of slopy fields. We wish we started from the top of the hill, but we didn't. Winter crops are the main crops that we grow, and those crops are mainly winter wheat. We can't really get in the fields in the spring because crops are growing and soils are wet, because we have pretty silky and soils, so it could be very muddy in the spring. So we really can't get in the fields in the spring. So we took soil samples in October after harvest, which is very different from most of you guys. So we took samples in October in 2018. So the specific presentation, this presentation that Catherine prepared really focused on looking at how abiotic indicators such as variable pools of carbon, nitrogen related with biotic indicators. Mainly she was looking at PRFA results. And she looked at the correlation between biotic and abiotic indicators across agroecological zones as well as across depths within each site, and see if there are any differences in those correlations in different environments, different fields, and different depths. So if you, so I just randomly put two figures in here looking, yes, thank you. Okay. So the takeaway is that abiotic indicators she was looking at carbon and nitrogen, and biotic indicators of soil health are significantly different at depth and those abiotic and biotic indicators are correlated, sometimes are very strong correlated, sometimes the correlation is weaker. So interpretation of those correlations, really it takes time, but it's interesting, and of course, there are some correlations we need to be very careful to interpret. We need to look into more and dig into more to be able to interpret them in a correct measure, scientific manner. So that's basically what we get. Please go ahead and if you have time and listen to our recorded video.
Yes, there's a lot of good information. Thank you, Hai-Ying.
Yeah,
thank you. Yeah, you guys had a very detailed sampling plan. Okay, we've got one more that I'm going to skip to, and that does not look right. Maybe I went the wrong way. Yeah, here we go. Okay, Cedric, are you on?
All right, thank you, Sky. Good afternoon, everyone. So our project is a fairly recent project that just got started this year, or in 2020, and it's a joint project between Alabama A&M University and the University of Maryland Eastern Shore. And because it's a new project, you can go to the next slide, Sky. Because it's a fairly new project, we're still in the planning stages, wrapping up the design, but again, sort of that goal is to assess the usefulness and the repeatability of soil health metrics on the Delmarva. And this is where the University of Maryland Eastern Shore, this is where they will play that role. Efforts so far have been sort of dedicated to getting up and going. That's why you see the redesign of an existing lab into just strictly a space for this soil health project. And then to accommodate hopefully future projects. But we have looked at, or they have looked at, site selection. And if you look there on the slide, it's probably not so clear right now. But we sort of got an idea of the distribution of crop, pasture, and hay within the state of Maryland and the Delmarva Peninsula, where University of Maryland Eastern Shore is located. So the next steps that they'll be doing is sort of working with the local NRCS to identify local farmers or area farmers who have these practices and coordinating that sampling with them. And then, of course, going on to do the work of collecting those patterns from the selected field sites. You can go to the next slide. Here at Alabama A&M, we're located in northern Alabama in what is called the Tennessee Valley region. So again, our goal with this is to assess the accuracy and the repeatability and usefulness of the standard soil health metrics across the Decatur Soil Series. The Decatur Soil Series is a benchmark soil for the state of Alabama. And like I said, for the region that we're in, which is the Tennessee Valley region, which you can see there on the right-hand side, that is probably the most extensive soil within this region. And when it comes to site selection, we can sort of pick from different, I guess, management practices. There are those here who still use conventional management practices, so you still see a good bit of tilling, in my opinion, that is going on. Cover crops are just not widely implemented, but you do have some farmers who have started implementing cover crops probably over the last 5 years. And before that, they've implemented no-till for the last 20. So it offers a good mix from which we can choose these management practices. And then another one that's of interest is in the state of Alabama, there's a push for expanding irrigation. So thinking about the management practices or the systems that we want to choose, maybe perhaps that irrigated versus non-irrigated row crops. Assessing soil health within these systems would be useful for the state of Alabama as they seek to increase the number of irrigated acres. So again, just like with the previous slide from UMES, our next steps, which we're sort of doing now, is to identify those field sites, identify the local farmers, and then coordinate the field sampling for this study, which hopefully we can get going in the fall. But that is where we're at on this project.
All right, thanks. You guys are just getting started, but I think it's going to provide some really great results. So that should be everyone now, John, that we've gone through. If you have any questions you want answered, put them into the Q&A pod. That'll make sure they're highlighted. If you put them in the chat previously, we'd still like to have another shot at them to get somebody to talk. I mean, just as we get transitioned here, this has been a really kind of wild project from my standpoint. We have learned so much at every phase, from planning to coordination between landowners, other parts of NRCS, our cooperators. It's a really a big idea, and it's a thing you really want to think about when putting these projects together. And there's been a lot of great chat I've seen going on that, yeah, soil health is variable, and by soil management system, region, it's complicated, and it's not a done deal yet. We still need more science to back things up. So, John, do you want to go through the questions we have thus far?
I can certainly do that, Sky, yes. So I've been taking down the questions from...
Oh, go ahead.
Go for it.
Oh, I was just going to say I might be able to help on a couple, but go ahead.
Great. I've been taking down the questions from both the chat and the Q&A, so we'll go through them. A few of them actually have been answered already. So in those cases, I'll probably either skip over them or we might come back to them if we have time. What I will say is that there's been some great discussion in the chat, so thank you all for that. And I mentioned this, but the pre-recorded presentations, there is a link available for you to go and explore those after the presentation. All right. So the first question we have from JB, and it's for the NC State presentation, so for Caitlin. And that question is, were the soil components in the study confirmed, or did you rely on the map unit name?
I'm not sure what he means by soil component, like the soil theory.
Caitlin, yeah, that's what he means, and I can help out on this one a little bit if you need it. Go ahead though. But he means the soil series, and the named map unit is named Cecil, but the component or the name of the actual soil series that was observed.
I believe one of the pits may have been Cecil, but once all of the actual lab testing, the mineralogy and things like that was done, I think they actually graded out to me Madison.
Yes, that is exactly what happened. Go ahead.
Yeah. But according to like Bud Soil Survey and soil maps, everything was sampled in Cecil.
Yeah, so that's one of those things where it's very interesting to work with some management and soil physics professors that were used to identifying the dominant component in the map unit and applying that as soil. But then once you have soil survey there doing characterization pet-ons, they're actually like, no, this is actually in Madison. And so when you see it in my presentations, I usually have it labeled as Madison because that's the actual most accurate soil series identification.
Thank you both for that answer. All right, I'm going to move on to the next question. So this is a question for all the presentations who have collected data so far. And Leslie asks, how many growing seasons have indicators been observed? And is this long enough to determine if these indicators indeed improve productivity? So I can send that to you first, Guy, if you have people you want to call on to answer that.
Sure. I mean, does anybody want to answer it? We won't all speak at once. But yeah, go ahead and send that to me, and I'll try to recruit some answers and maybe go to the next one for now.
Sure. We'll let everyone think on that. So this one is a great question. This is for all the students who participated in presenting today. Just an open question, are any of you interested in working with NRCS in the future, either as a cooperator or potentially as an employee?
So here, this is Haiying. I am not a student, but my students, my PhD student indicated that she wants to. Catherine is working. She is a postdoc with me at WSU, but right now she is working with Pacific Northwest National Lab, and then she's going to graduate in summer 2012 next year. So, but anyway, so are we still discussing the previous question? Oh, sorry, I should let other students talk before I talked about the previous question.
Thanks for that answer, Haiying. Yeah, let's see if I can answer that.
Hi, this is Regina. I can jump in.
Go for it.
Yeah, so throughout this project, I've definitely been thinking about possible career paths at NRCS, and I couldn't say what that would give any specifics about what I would be looking for, what type of position I would be looking for. I'm still a few years out from graduating, but it's kind of occurred to me over the last couple of years working on this project that there's a lot of positions at NRCS that would check a lot of kind of boxes for me as far as things I look for in a job, working outside sometimes, getting to work kind of in soil science education area, things like that, that I think it could potentially be a good fit.
Thanks for your answer, Gina. Just seeing if any other students had anything they wanted to say to that question.
I think Sharon might want to jump in. I'm not sure if she's talking about this question or the previous one.
Sorry, you're looking for me to interject?
Sure, go ahead.
Oh, yeah, I was just wondering if any of these research teams have thought about working with the local NRCS soil surveyor field offices to incorporate their knowledge into our inventories, because as they may or may not know, our ecological site descriptions really need that scientific basis to help them become legitimate repositories for this knowledge. For how do we go forward using state and transition models to guide us in our land management decisions and our conservation practices if we don't have the fundamental science to back it up? And with the tens of thousands of ecological site descriptions out there, we don't expect there to be tens of thousands of scientific studies that go with each ESD, but we do need a basis, and we do need a foundation. So in order to start building that, we need to have that good cooperation between the academics and government institutions to have that synergy and to make these products really function through time. So I don't know if anybody wants to speak on that. I'd appreciate hearing what other people's thoughts are. Thank you.
I'd like to speak on that if you don't mind there, John.
Yes.
This is Kirsten from the University of Arizona. Yeah, so we actually have been working quite closely with our Tucson field office folk, particularly from Carrillo and Caitlin and Bill Stetlick before we unfortunately lost Bill to my ties probably. But yeah, it's one of those things that we've actually found really, really helpful from our side about the program, was the ability to have the expertise of NRCS field staff, particularly with the PDON descriptions and understanding how soil survey links in with soil health assessment. So, but I will say that initially, and this is through no fault of the Tucson field staff at all, that it was a little bit difficult to understand how the roles intertwined. And so I do agree with you that that is something that really needs to be further developed so that we can get the most out of these programs for both parties, for the academic side and also for the field office side.
Excellent. Thank you for that answer, Kirsten. Did any other presenters want to comment on that question as well? Okay. I'll just backtrack very slightly to wrap up the question on student interest in working with NRCS. I'll say that our recruitment, leadership, diversity, and recruitment focus team leader, Leslie Glover, asked that question. So we do have resources for those students who are interested to provide more information about jobs that will be available this year. So please look forward to that. I've taken down the names who have expressed interest. Sky, do we want to return to the growing seasons question? I see some answers coming out about that.
Yeah, why don't you read that question again, and we'll chime in.
Perfect. I will read it again. All right. So the question is, how many growing seasons have indicators been observed, and is this long enough to determine if these indicators indeed improve productivity?
So I will start by saying that these indicators were chosen because they change relatively quickly, but they're meant to be relatively stable across seasons and conditions, so that we don't expect them to change. Like, we're not implementing a change for a year and then measuring. And for the most part, we're just using found management systems and measuring those, so it varies quite a lot by project, how many years those management systems have been in place. And so if anyone wants to talk specifically about their system and whether they think the indicators will be related to productivity because of how long those systems have been in place, it would be great to hear from you. But it's going to vary by project.
I can talk about our site for a little bit. So we had two sites that we looked at. The first one was in Bushland, Texas, and that site was established in the 1970s, large-scale plots. The second site was the Amarillo series in La Mesa, and that one was established in 1998, and these have been consistent research plots. And what we've seen is that there was not a relationship between soil health indicators, specifically the biological indicators, and yield, which is what most of our growers are most interested in with cotton production. We started measuring these soil health indicators, specifically the biological indicators, in 2015, and then the results that I showed today were from 2018. So there were 3 years that we looked at, or 3 growing seasons, particularly for that project.
Thank you so much. Any other comments presenters want to make?
This is Haiying Cao from Washington State. I just want to talk a little bit about the statewide effort from Washington State. So the Washington State has been very interested in soil health issues that we have in Washington State. So the state actually initiated a soil health initiative 2 years ago, but we got the first year money and the second year due to COVID, so this thing is at a stop right now. But so one of the things that this effort is starting doing is to take soil samples from growers' fields, from different soil types, different cropping systems, different soil management systems, basically to do a broad survey and to establish baseline as well as to look at if, pretty much similar to the overall objective of our project with that we're presenting right now. So, and again, it's ongoing. So, and specific, one of the specific project I want to talk about is that I think really depends as far as how we correlate the soil health indicators to the yield, productivity, or how well those indicators are correlated with those yield and quality and productivity. Really depends on the parameters, I think. One of the things that we have to deal with in dryland cropping systems in no-till field is a pH issue. So we have severe soil acidification problems in no-till soils, and absolutely, you see a strong positive linear correlation with pH and yield. And then you dig deeper. What is pH doing in the soil as far as soil fertility, soil microbial health, and soil physical health, and then how we correlate each of them to the yield and quality of the crops as well as productivity. So I hope I'm not confusing people by jumping around with my thoughts.
No, that's great, Hai. I think that's good to connect to the different questions that we're being asked with the work you're doing. So thank you. Just making sure that if there was any other presenters who wanted to comment on questions about growing seasons and linking indicators to improve productivity.
This is Carmen Ugarte, and I wanted to comment on what we've been finding also in relationship to the indicators and productivity. We've been looking at several different indicators of bioavailable carbon and nitrogen, and we're finding that there is more than one factor or property that we should be looking at. And we think specifically on carbon and nitrogen. In my opinion, we should also be considering drainage class and weather, and try to integrate multi-set of variables or do some multivariate analysis type of thing so that we can better relate it to yield. We've seen also the stories of phosphorus and potassium from the past, and despite the fact that we can quantify it pretty well, it's still hard to make or to find direct relationships with yield. So that's my 2 cents.
Thank you very much for that. All right. Any other comments before we move on to a new question?
Yeah, this is Chuck Rise. Can you hear me okay?
I can, yes. Please go ahead.
We've actually started, it's not part of this NRCS funded project, but another one that we're looking at, spatial variability of soil health related to implementation of soil health improving practices like no-till and cover crops. And so we just started, we got baseline, and then after 3 years, and we're tracking that. So we are starting to see trends for improving some of the soil health metrics. And then one of my students has correlated that with spatial maps of corn and soybean yields. And as one of the other people commented, we've looked at kind of elevation and wetness index. And so what he found was that the soybean yield wasn't necessarily correlated with some of the soil health practices, but corn, or metrics, but corn was correlated. And organic matter, we're seeing some changes, early indicators, trends, not significant yet, but after 3 years of improved aggregation. And there's also a relationship in the aggregation and the amount of microbial biomass as well. So, you know, we're just at the cusp of maybe seeing significant differences, but that's what we're seeing so far.
Thank you for sharing that, Chuck. All right, another call. Any other comments on growing seasons and indicators relationship to productivity? All right. Well, thanks everyone for answering that question. I also said that we had a similar question about relationship between soil health and improved productivity. So I think that we were able to, a lot of you were able to address that as well. Not many more questions, so attendees, if there are more questions you have that have come up, please put them into the Q&A. We still have as many as 9 minutes left. And while we're waiting for more questions, I did get one in the chat from Aaron. He says, it's good to see folks collecting baseline data for future observation, but the question is, how long does it take to return to pre-plow conditions? So that's quite a big question, so if anyone feels up to answering that right away, please go for it.
John, this is Sky. I'm not going to say how long it takes because I think it's very dependent on climate. It really is. But we are collecting these with the idea that they serve as reference conditions, and that's part of why we try to get a reference condition to measure in addition to these, you know, business as usual and kind of soil health management systems, is so that we have an idea of sort of the range we might expect, what's typically there, and then what the reference conditions might be. And that at least lets us know in the future as we make more measurements how those compare to what's possible. So I don't know if anybody wants to speak to their specific area or conditions.
Yeah, Sky, I'll jump in here. And Chuck Rice, we have one experiment that we've been going for 33 years of no-till, tilled, and then comparing inorganic and organic fertilizer nitrogen sources. And then we also have a reference site for prairie, tallgrass prairie. Again, we're at 33 inches of rainfall, 850 millimeters of rain. So there's two parts of that question. One is, or two answers. One is that with no-till and adding compost for 33 years, we're getting carbon, I would call it saturation, equilibrium, depending on who you want to talk to, at the surface. We got aggregate numbers that are getting close to what the native prairie was, but not there yet. But the other question, I guess, a response, is it really, if we're talking a perennial system, is it really fair to compare an annual versus perennial? They're completely different systems, root depth, productivity in that sense. So I'm not sure you would expect or should expect an annual system, even with cover crops, no-till, all those components are ever going to meet what like a prairie or forest would have been.
I appreciate that answer. Thank you. All right. If it's all right, I think I'll move on to the next question. Just checking unless any other presenters wanted to comment on that. Okay.
This is Heidi. Sorry. Yeah,
oh, yeah, go for it, Heidi.
So I just want to add, so really, I mean, whether you can go back to pre-plow really depends on what parameter you are looking at. For our soils, we we had really, really bad erosion in no-till, in the conventional tilled systems. We lost a significant amount of topsoil. If you want to go back, if you think about it, okay, how to go back to pre-agriculture condition is almost impossible unless you bring the topsoil back. But, if you could build up organic matter by better management practices.
Excellent. Thank you for that additional answer. Appreciate it. All right. As we have 5 minutes left, if anyone has questions they want to submit now, please do. If we don't get to any questions, we will try to address them after the webinar with a written question and answer. All right. I do have a question for Sky, and this is specifically asking, can you comment on whether or how DSP metrics might be revised or replaced, i.e. those that are potentially too time intensive or end up not being relevant or useful for soil health assessments? So if you're ready for that, Sky, that's a question for you.
Yeah. Yeah, and it's actually something we think about a lot, right? Because this whole project was bridging soil survey and what we consider dynamic soil properties and we measure, which are usually meant to be things that change on the decadal time scale, that are reliable, that aren't too variable, and don't vary too much with like daily conditions. And we also tested things that were in the Soil Health Division's Techno, which are these indicators by functional groups, or functional areas, I should say. And I am not sure what their update process is, but some of the metrics are pretty standardized because lots of people have done them in lots of places. I am sure that we will continue to tweak these methods. I am less sure about what the timeline is on when those become quote-unquote official, because this is still a developing area. I wouldn't say that any of these are really written in stone.
Thanks for that answer, Sky. All right. Well, at this time, we don't have. Be safe out there, and have a good rest of your day.